Hello, my dear students. Welcome to my today's online class on one more technique often used in biotechnology, that is RFLP, which stands for restriction, fragment, length, polymorphism. Okay. So this is a slide giving my introduction that I am Dr. MEERA B K, Associate Professor and Head, Department of Zoology, Maharani Science College for Women, Palace Road, Bengaluru 1. You can see my cell phone number and mail ID on this slide. After the class, any queries, do contact me over this. Now, so the diagram what I have put here will tell you the schematic happenings during RFLP. Okay, what all you are going to learn in next coming few minutes, all that has been summarized here. It is just a schematic representation of RFLP. Okay, now therefore I have highlighted this. Then what is RFLP? A RFLP represents a stretch of DNA that serves as a marker for mapping a specified gene. So basically, it is a stretch of DNA that serves as a marker for mapping a specified gene. Though definition wise it goes like this. Let us just try to understand in simple terms what exactly RFLP means. There are 4 terms in this. Restriction means the DNA fragments which have been subjected to the digestion of restriction endonucleases, and whatever the DNA fragments you get after the restriction digestion, that they are called restriction fragments. Okay. Then its length. So the length of restriction fragments after digesting the DNA by using restriction endonucleases, and their polymorphism. Poly means many, morpha means appear, many forms. So in simple terms, you can take a genomic DNA and treat them with restriction endonucleases. Okay. So if there are n number of target sites in the genome, after restriction digestion, you will get n plus 1 number of DNA fragments. Okay. So they are restricted fragments, and their length varies from one fragment to another fragment depending upon the place of the presence of target site. Therefore, you get many forms, polymorphism. So that's what I exactly meant by restriction fragment length polymorphism. Okay. See, normally these RFLPs are located randomly throughout a person's chromosomes, and apparently they don't seem to have any functions. Now, you can cut a DNA molecule into different fragments by restriction endonucleases, and whatever the fragments outcome you get are called polymorphs. Poly means many, morph means forms, polymorphs. Okay. Now, let me explain this diagram. So, the diagram explains you an outline of RFLP. Okay. So what exactly it tells us? Let us see. So this is a DNA molecule. There are 2 DNA molecules, DNA 1 and DNA 2. You can make out in the diagram that DNA molecule 1 has 3 restriction sites, R1, R2 and R3. Alright. And when you cleave them with restriction endonucleases, you will get 4 fragments. I had already told you that you get n plus 1 number of fragments, where n is the number of restriction sites. Here there are 3 restriction sites, n is equal to 3. So n plus 1, the outcome is you will get 4 fragments. Okay. This is the fate of DNA molecule 1 after subjecting it to restriction digestion. Now you can see the bottom part where we have another molecule, DNA molecule 2. For our understanding, we have called it as DNA molecule 2. The difference is the restriction site 2 is missing. Restriction site 2 is missing. There are only 2 restriction sites. So when you subject this to restriction digestion by making use of friends, okay, how many fragments you get? You will get only 3 fragments. Right? So let us consider that this DNA molecule 2 has an inherited mutation. Okay? Has an inherited mutation or a genetic change, and therefore has altered some base pairs. And as a consequence of this, the restriction site R2 is lost. So when you compare the products of DNA 1 and DNA 2, you can always say that there is a mutation in DNA molecule 2. This is what exactly is done in RFLP. This is how the analysis is done. Thus, a stretch of DNA exists as fragments of various lengths. That's what I meant by polymorphism, derived by the action of restriction enzymes. So hence the name RFLP. Okay. Now, same thing I have explained here. DNA 2 with an inherent mutation, therefore altered base pairs, and as a result R2 is missing. Therefore, when it is cut by restriction endonuclease, you get only 3 fragments instead of 4 as you see in DNA molecule 1. So this is what exactly happens in RFLP, and this is how analysis is done. When you look at DNA 1 and DNA 2, you can always say that in DNA molecule 2, there is some inherited mutation. As a result, restriction site 2 is lost. Okay. Then, so this is the dogma. Restriction site 2 is lost. Right. Then, there are 2 methods of detection of RFLPs. One, you normally do it by southern hybridization. Okay. What is done in southern hybridization? Basically, what is southern hybridization? It is a nucleic acid hybridization technique. Okay. So in this southern hybridization, the DNA is digested with appropriate restriction endonuclease. You will get DNA fragments, and you can always separate out these DNA fragments by subjecting them to electrophoresis. And the electrophoresis type what we use here is H, agarose gel electrophoresis. Then the so obtained DNA fragments after H or after subjecting it to agarose gel electrophoresis, you can shift these DNA fragments to a nylon membrane. Okay, nylon membrane. Then a DNA probe that spans the suspected restriction site is now added, and the hybridized bands are detected by autoradiograph. I'll show you the diagram later in the next slide. So what you will do, you will take the DNA, digest it with appropriate restriction endonuclease, and then you will subject it to H. You will get so many DNA fragments that you will transfer it to a nylon membrane. And then you will add a probe. You will add a probe that spans the suspected restriction site. And hybridization takes place. And these hybridization bands, normally in southern blots, are detected by another technique called autoradiograph. I'll give you one homework. You can just make a note of your autoradiograph technique, okay, and keep as a supplementary reading to your reading on RFLP, okay. Then, if the restriction site is absent, say for example inherited mutation or something else, then you will get only a single restriction fragment, okay. On the other hand, if the site is present, you will get two fragments. This is what happens during southern hybridization. Let us see the slide now. You concentrate on the figure A, okay. So the common methods used for RFLP analysis, this method A is by southern hybridization. You can see here a DNA piece containing three restriction sites, R1, R2, R3, right? DNA with restriction sites. Now for this what you will add? Restriction endonuclease, and then you can see at the place of R2 you have put a probe, right? DNA probe, right? Then the cut ones, the DNA fragments have been transferred to nylon membrane, and that suspected R2 site DNA probe is added, and it will hybridize with the bands here. So if restriction site is present, you will get two bands as you see here below. If the restriction site is absent, you will get only one band. This is how you will analyze the RFLP through the southern hybridization technique, okay. So let me summarize here. The DNA is digested with appropriate restriction endonuclease and then subjected to age. The so obtained DNA fragments are transferred to the nylon membrane, and then a DNA probe that spans the suspected restriction site is now added, and the hybridized bands are detected by autoradiograph. If the restriction site is absent, you will get one band as you are seeing in the diagram or the autoradiogram. Whatever you get, outcome of autoradiography is autoradiogram. And if the site is present, you will get two bands. This is how the RFLP is done through southern hybridization. Another method by which you can do is PCR, polymerase chain reaction, the technique I have taught you in my last class. Let's see what is done in this. For this, PCR primers that anneal on either side of the suspected restriction site are used, okay. Then, after amplification by PCR, the DNA molecules are treated with restriction endonuclease, same step, and then analyzed by agarose gel electrophoresis, same step. If the restriction site is absent, only one band is seen. If restriction site is present, then you can see two bands. So same slide I have put here so that you can concentrate on the diagram B and compare with A. Okay, A for southern hybridization, B through PCR. Here you can see the same DNA piece are with restriction sites R1, R2, R3, and you are suspecting R2, right? Suspected site is R2. You will put PCR primers on either side of the DNA, okay, with restriction map. Then you will run the PCR. Then you will treat it with restriction endonuclease, then subject to agarose gel electrophoresis. Then you will go for hybridization, and if there is one band, it tells you that suspected site is not there. If you see two bands on autoradiogram, it tells you that suspected site is present, okay. So this is what is done in RFLP. Now let us see the applications. If the RFLP, that restriction fragment length polymorphism, lies within or even close to the locus of a gene that causes a particular disease, it is possible to trace the defective gene by analysis of RFLP, okay, in DNA. So what you have to do is, the person's cellular DNA is taken isolated and treated with restriction endonucleases. Then the obtained DNA fragments are separated by age. The RFLP patterns of the disease suspected individuals can be compared with same of normal people. So you have to repeat what all you have done for a suspected person, all that you have to do for a normal person, preferably the relatives in the same family. Okay. Then you can compare the band patterns of a suspected deceased person and a normal person. You can say whether the person has the disease or not. By this approach, it's possible to determine whether the individual has the marker RFLP and the deceased gene. Okay. So with 95% certainty, RFLPs can detect single based diseases. There are few success stories. Okay. And this approach is quite powerful and has helped many genes to be mapped on the chromosomes. For example, sickle cell anemia. They have come to know that it is on chromosome 11. Same way, cystic fibrosis on chromosome 7, Huntington's disease on chromosome 4, retinoblastoma on chromosome 13, Alzheimer's disease on chromosome 21. Okay, they are all single gene based diseases, and they have been able to spot out this particular position of these genes through RFLP. Okay. Now you can see this slide. Here is the DNA piece, okay, with restriction sites for MSTL, okay, and then this is another piece, right? So this is from normal person. This DNA is from normal person, preferably from the same family. This DNA fragment is from the deceased person. Now there are 3 restriction sites, and if there is a mutation, say for example, this mutation would have destroyed one restriction site. Now what all I explained, you have to do that. Treat with run, then run on AGE, then hybridize, right? So if you put them on age or gel electrophoresis, the band patterns you can see the normal and also disease. You can make out that particular few bands are missing in the deceased person, right? In the deceased person, especially the band here. So in the Southern blot analysis, you can compare the normal person's blots and the blot of deceased person. This is how exactly the bad genes are located. Okay, and you can just say by RFLP analysis whether the person carries a bad gene, whether the person will develop the disease. Okay, this is all about RFLP technique. My dear students, thank you for your patient listening. Any queries, do contact me. Stay blessed. Thank you. In my next class, I am going to teach 2 more techniques like this. One is RAPD and one is AFLP. Then we shall have a comparative account of all the 3 techniques, RFLP, RAPD and AFLP. Till those 2 techniques are over, you can just concentrate the reading of this technique. Stay blessed. Hello, my dear students. Welcome to my today's video lecture or online class on RAPD. RAPD means random amplified polymorphic DNA. In the last class, you have learnt about RFLP, and this is another technique which is available, which is in use right now, especially in the field of plant taxonomy. This RAPD is also called arbitrarily primed PCR, arbitrarily primed PCR, ATPCR. I'll let you know why it is called so little later. I think I'll skip this slide. By now you know who I am and why my mobile number and mail ID are there on this slide. And you can see one more picture which I am going to explain it a little later, that gives the schematic representation of RAPD. Okay, that's the reason why I have highlighted it. Now, normally in PCR, what I have taught you in my previous lectures, the main objective is to generate defined fragments of DNA from highly specific primers. That's what we did in PCR. But what are we going to do in RAPD? See, in RAPD, short oligonucleotide primers are arbitrarily selected to amplify a set of DNA fragments randomly distributed throughout the genome. Right now, you just remember this. When I explain the technique with the schematic representation, you will understand it better. So, here you make use of oligonucleotide primers which are arbitrarily selected to amplify a set of DNA fragments randomly distributed throughout the genome. Hence, RAPD is also called arbitrarily primed PCR. This is why it is called APPCR. Okay. But overall happening in RAPD, that is procedure, is comparable to the general technique of PCR. This method involves the use of a single primer at low stringency. Okay, let's see what exactly happens. Now, I told you the process is almost similar to PCR, but what are the differences or what are the comparisons? This slide will tell you the components of a PCR reaction and the components of an RAPD reaction. See, in both the cases, the buffer contains magnesium ions. But in RAPD, normally you give higher concentrations of magnesium ions. Why? For lowering annealing stringency. Okay? For lowering annealing stringency. So in RAPD, you give higher concentrations of magnesium ions compared to PCR. What is the template used? In both the cases, it is DNA. In PCR, as you have already learnt, two primers that flank the fragment of DNA to be amplified is seen. You make use of two primers which flank the region of the DNA to be amplified. Please note, but in RAPD, you make use of one short primer, normally 10 bases length, and not known to anneal to any specific part of template DNA, right? Randomly it goes and attaches to different regions of genome. This is the major difference between a PCR reaction and a RAPD reaction. Of course, in both you have to give DNTPs, that is ATP, GTP, CTP and TTP for furtherance of amplification. Then in both cases you make use of Taq polymerase, that is thermostable polymerase, DNA polymerase, or any other thermally stable DNA polymerase. So if you are asked to write, compare and contrast a PCR reaction and an RAPD reaction, these are the points which you are going to highlight. Then, what is the principle of RAPD? What exactly is done? See, in the diagram you can see the DNA template, and that you are going to subject it to RAPD. You are going to put the primers randomly, right? And then the PCR reaction will take place. So you will get RAPD PCR products. you can see the two bands, two amplified DNA pieces, and that you can always separate out on gel electrophoresis, and you will get the banding pattern as shown in the diagram. So detection of the size of the amplified fragments is done, and therefore you can always compare. This is the basic principle of RAPD. Okay. So a short oligonucleotide primer of usually 9 to 10 base pair binds to many sites in the genome, and the DNA fragments are amplified from them. Then the stringency of primer binding can be increased after a few PCR cycles, but initially in RAPD you have to keep stringency little lower only, and that is done by keeping a higher concentration of magnesium ions, right? Then when this happens, amplification of best mismatches will take place. That's the reason. Then RAPD can be carefully designed so that it finally yields genome-specific band patterns that are useful for comparative analysis. This is possible because genomic DNA from two different individuals often produces different patterns by RAPD. I'll show you the diagram a little later. You will understand this point better. Thus, a particular DNA fragment may be generated for one individual and same may not be generated for the other. And this represents DNA polymorphism, and that can be used as a genetic marker. So this is the platinum sentence, okay, in your answer. Therefore, I have highlighted. I'll repeat. A particular DNA fragment may be generated for one individual, and the same may not be so in another individual, and therefore there occurs DNA polymorphism, which can be used as a genetic marker. So this is the schematic representation highlighting the different steps. See, here we have two samples, sample A and sample B. We have isolated the genome or the DNA. What you are going to do is, you will give them DNA polymerase. Of course, by now you know it is Taq polymerase or any other thermostable suitable polymerase. And you will give short universal primers. Please note. Okay, you don't give two primers which anneal to the flanking regions of the DNA on either side. What you do in PCR? No, not at all. You will give short universal primers, oligonucleotides of 9 to 10 base pairs length, and you will conduct the PCR. You can see the figure A will show you the PCR reaction for sample A DNA, and figure B will show you the PCR reaction for sample B DNA. Then once the amplification is through, you will put them to electrophoresis, and you can see the banding patterns developed for sample A and for sample B, and definitely there is polymorphism. Okay, this is how you can make use of RAPD as a marker. Okay, in this scheme, the dotted lines will tell you that they are the primers, and the thick dark lines are the amplified fragments, and arrow mark will show you the direction of amplification. This is what is exactly done in RAPD. It is widely used by molecular biologists for genetic identification of plant species. So this has a wide application in molecular plant taxonomy. Okay. For this purpose, what they do is combinations of nucleotides. Most of them are oligonucleotide primers, as I have already told you. They have been designed and they are readily available commercially. Okay, simply you can go buy the primers of your choice and do RAPD. As each random primer anneals to a different region of DNA, many different regions of loci on DNA can be identified. Okay, and therefore RAPD is useful for the construction of genetic maps, and it also serves as a method for genomic fingerprinting. So the two major applications of RAPD are number one, construction of genetic maps, and number two, they serve as method for genomic fingerprinting. With all this RAPD, though much better than RFLP, has its own limitations. The main problem is associated with reproducibility. Okay, it is often difficult to obtain similar levels of primer binding in different experiments, and therefore difficult to correlate the results obtained by different research groups on RAPD. This is the major limitation of RAPD. This slide will just compare the RAPD and RFLP techniques. Let us see one by one. I'll just zoom it. In RAPD, it is basically a PCR based technique for identifying genetic variation. But RFLP is a molecular method of genetic analysis which allows individuals to be identified based on unique patterns of restriction enzyme cutting in specific regions of DNA. So this is PCR based method and this is restriction digestion based method. Then RAPD involves amplification of genetic markers in a genome by using random primers, whereas RFLP involves restriction digestion of the genomic DNA by making use of restriction endonucleases. For RAPD, small quantity of DNA, 10 to 50 nanogram is enough for the analysis. On the other hand, RFLP requires comparatively large quantities, okay, of DNA, 2 to 10 micrograms. Here it is nanogram, here it is micrograms, okay. Then RAPD uses random primers universal for any species as they are readily available commercially, whereas RFLP uses different species specific probes. Then RAPD has comparatively fewer steps and therefore it is a rapid process, whereas RFLP has more steps and therefore it is a slow process. RAPD is less reliable whereas RFLP is more reliable. Okay, this is a difference. Then RAPD can detect 1 to 10 loci, whereas RFLP can detect only 1 to 3 loci. Then RAPD is not able to detect allelic variants. Allelic variants cannot be detected by RAPD, whereas RFLP can detect allelic variants. Allelic variants. Okay. So these are the differences and comparisons between RAPD and RFLP. Okay. My dear students, thank you for your patient listening. Any questions, you can contact me either over my cell phone or through mail ID. Thank you for your patient listening. Stay blessed. Thank you. My dear students, welcome to my today's online class on amplified fragment length polymorphism. And by abbreviation, it goes like this, AFLP. You have learnt RFLP, RAPD in my previous classes. Now, we shall learn one more such technique, and at the end try to compare and contrast all the three techniques that have been discussed so far. So today we are discussing about amplified fragment length polymorphism, AFLP. So I am Dr. MEERA B K, Associate Professor and Head, Department of Zoology, Maharani Science College for Women, Palace Road, Bengaluru 1. You can see my cell phone number and email ID, and after the class any queries you can contact me. Now you can see one more picture, which is the schematic representation of the technique what we are going to learn today. Okay? Yeah, this is the reason why I am highlighting it. AFLP is a very sensitive method for detecting polymorphism in the genome. Same job is done by RFLP, same job is done by RAPD, but of the three, AFLP is quite sensitive method for detecting polymorphism in the genome. It is based on the principle of RFLP and RAPD. In a way, it is a hybrid of RFLP and RAPD. Okay? It is appropriately regarded as diagnostic fingerprinting technique that detects genomic restriction fragments. I have not taught you DNA fingerprinting so far. In my next coming classes, I am going to teach that. Right now, you just remember that AFLP is treated as a diagnostic fingerprinting technique, and what it does, it detects genomic restriction fragments. Here, we make use of PCR amplification rather than Southern blotting for the detection of restriction fragments. By now you know that in RFLP, we learned that for scoring both PCR and Southern blotting are used, but in AFLP, we make use of only PCR amplification and not the Southern blotting. This is a difference between RFLP and AFLP. Very important thing to be noted is AFLP is employed to detect the presence or absence of restriction fragments, please note, and not the lengths of these fragments as done in RFLP. This is the major difference between AFLP and RFLP. I repeat, AFLP is employed to detect the presence or absence of restriction fragments, not the lengths. Okay, so that makes the technique quite sensitive. All right. So since it is very important, I have highlighted this way. AFLP is widely used in plant genetics rather than animal genetics. I'll let you know why it is. See, normally AFLP is not used for mapping animal genomes because this technique is mainly based on the presence of high rates of substitutional variations which are not found in animals. But such substitutional variations resulting in RFLPs are more pronounced in plants. Thus, AFLP finds applicative value more in plant genetics rather than in animal genetics because substitutional variations are normally not found in animals. This is a point to be noted. The basic principle of AFLP is the amplification of subsets of RFLPs using PCR. Alright, let's see what exactly is done. So, in this slide, I am going to take a particular example and explain the technique. So, see this schematic representation. What all is being done, let us see. A genomic DNA is isolated and digested simultaneously with two different restriction endonucleases. You can see here is the genomic DNA. On one side you have the sequence GAATTC, which is the recognition sequence for EcoR1. On the other side you have the recognition sequence TTAA, which is the recognition sequence for the restriction enzyme MSEL. Thus, EcoR1 has a recognition sequence with a 6 base pair, whereas MSEL with 4 base pairs. Now, this is the genomic DNA, and here is one site for EcoR1 and one site for MSEL. Both of them are restriction endonucleases. Now treat this DNA with both the restriction endonucleases simultaneously. Then what happens? These two enzymes will cleave the DNA and result in small fragments, small DNA fragments which are less than 1 kb, so that they can be amplified by PCR. So in the second step you can see you will get the restriction digested products. Alright. Then for this purpose, to run the PCR reaction, the DNA fragments are ligated with EcoR1 and MSEL adapters. Till now, I have not taught you about linkers and adapters concept, which are often used in recombinant DNA technology. See, once you digest the DNA with restriction endonucleases, there comes a concept called modification of cut ends of DNA. There are around 10 methods by which you can modify the cut ends. By now you know that they produce either blunt ends or 5' overhangs or 3' overhangs, depending upon the way the restriction endonuclease cleaves the DNA. I have taught all this in my previous lecture. Now, once you get the cut ends like this, you can always modify the cut ends so as to suit to your experiments. There are around 10 methods, and out of 10, one such method is adding the linkers and adapters. Okay, so just right now you can just know that you will add here EcoR1 adapter and MSEL adapter. Adapters can be designed for different restriction endonucleases, and accordingly you can use whichever adapter you want for your experiment. Okay. So to run the PCR, before running, you have to modify the cut ends, for which you have to ligate the DNA fragments with EcoR1 and MSEL adapters. See, here is the case. This side I have shown EcoR1 adapter, and this side I have shown MSEL adapter. Right? They can be designed in the laboratory. Then, these common adapter sequences, flanking genome sequences, serve as primer binding sites on restriction fragments. Why do you add adapters? To serve as primer binding sites on the restriction fragments. You know, PCR can't be carried out without the primers being binding to the DNA fragments, right? Then, the DNA fragments can be amplified with AFLP primers. each having only one selective molecule. In the initial stage, you will add first primers with one selective nucleotide, okay, one selective nucleotide, and you will get the PCR products, and these PCR products are diluted, and again they are used as templates for selective amplification, employing two new AFLP primers that have 2 to 3 nucleotides. This is the next step, right? Addition of first primers with one selective nucleotide. Once you get PCR product, then addition of second primer with 2 to 3 nucleotides, okay? Then after the selective amplification by PCR, the DNA products are separated on a gel. I'll show you in the next slide. The resultant DNA fingerprint is identified by autoradiography, and these AFLP fragments represent unique positions in the genomes and hence can be used as landmarks to bridge the gaps between genetic and physical maps of genomes. Okay. In plants, AFLP is useful to generate high density maps and to detect genomic clones. So whatever I have shown in this slide, I'll just repeat. A genomic DNA is isolated and digested simultaneously with two restriction endonucleases, EcoR1 and MSEL. Okay. Then the two enzymes will cleave the DNA and result in small fragments less than 1 kb, which are amplified by PCR. To do this, the DNA fragments are ligated with EcoR1 and MSEL adapters. These common adapter sequences serve as primer binding sites on restriction fragments. The DNA fragments can be amplified with AFLP primers, each having only one selective nucleotide first time. Then whatever PCR products you get, they are used as templates for selective amplification. Employing two new AFLP primers that have 2 to 3 selective nucleotides. Okay, that is the next step. Then after the selective amplification of PCR, selective amplification by PCR, the DNA products are separated on a gel. The resultant DNA fingerprint is identified by autoradiography, and AFLP fragments represent unique positions in the genomes and hence they can be used as landmarks to bridge the gaps between genetic maps and physical maps of genomes. Okay. So in plants especially, this is useful to generate high density maps and to detect genomic clones. Okay. Let us see in next slide. Same thing whatever I explained, I have put it here for your reference purpose as well as for reading purpose. This serves as a study material. Okay. Same thing. So 2 to 3 nucleotides, then you will run on a gel by autoradiography. Now, again this is a very important concept what I have stressed already, right? They serve as landmarks to bridge the gaps between genetic and physical maps of genomes. Then you can see this schematic representation. What all I have explained with one particular instance, same I have given a general schematic representation. You will isolate total genomic DNA. First step, you will conduct restriction digestion. You making use of restriction enzymes. You will get so many DNA fragments. Then you will do adapter ligation by putting the adapters which are designed for particular restriction endonucleases. Then you will do pre-amplification Then you will do selective amplification, and whatever fragments you get, that you will subject it to gel electrophoresis. And this slide gives you the overall picture of what is done in AFLP. Yeah, this is another schematic representation briefing you about the various steps involved in AFLP. So genomic DNA, you put restriction enzymes for digestion, then you will give adapters for ligation, then you will get ligated DNA fragments, you will amplify them, right, selective primers, and you will get amplified fragments, and you will run on polyacrylamide gel. Okay, there are two types of gel electrophoresis: age agarose gel electrophoresis and page polyacrylamide gel electrophoresis. In this case, you have to run page, and you will get the bandings like this. Here they have taken 3 samples of DNA, A, B and C. Look at the fingerprint pattern. You can make out that sample B and C are almost same fingerprint pattern, whereas the pattern obtained for sample A is different, right? This is how AFLP is done. Now this slide briefs about RFLP versus RFPD versus AFLP. In RFLP, it is a codominant marker. RFPD is also a codominant marker. AFLP is a dominant marker. RFLP, non-PCR based approach. RFPD, PCR based approach. AFLP, combination of both RFLP and RFPD. RFLP, there is need for restriction endonucleases. RFPD, no need of REN, whereas in AFLP, there is need of restriction endonucleases. RFLP requires large quantities of DNA. RFPD, very meager amounts of DNA. And AFLP do not require large quantities of DNA. Then RFLP primer is not needed because this is a non-PCR based approach. RFPD, it uses multiple arbitrary primers and lead to unreliable results. That is one drawback of RFPD. Whereas AFLP technique uses only 2 primers like PCR and gives reproducible result, reliable result. Then in RFLP, no such rule to use restriction enzyme. Anything you can use. RFPD, you don't use restriction enzyme at all. AFLP, combination of restriction enzymes are used. Okay, frequent cutters and rare cutters. So this slide briefs you the RFLP versus RFPD versus AFLP. Right? It gives a comparative account of the 3. Then we have to discuss advantages and disadvantages of these 3 methods, whatever you have learnt. Coming to RFLP, advantages: paternity test, it is used for identification of criminals in forensic medicine and to identify genetic diseases. Disadvantages: it is slow, more tedious, quite costly and requires more DNA samples. On the other hand, AFLP is used, has advantage: positional cloning of gene of interest, high reproducibility, then many loci can be analyzed simultaneously. Disadvantages: complex procedure, non-identical commigrating bands can cause noise. That is one problem. RFPD, genetic relation establishment, genetic diversity studies, analyze mixed genome samples, applicable when limited DNA is available, works out economical. Disadvantage: markers are dominant, that is, they can't distinguish whether DNA sequence is amplified. DNA sequence amplified is heterozygous or homozygous. So these are the advantages and disadvantages of RFLP, AFLP and RFPD what you have studied. You can see one more marker here, SSR and ISSR. So they are also a type of markers. I have not taught you because this is not there in the syllabus. So dear students, till now I have explained you the AFLP technique and I have given a comparative account of all the three techniques whatever you have learnt, and have also highlighted the advantages and disadvantages of the three techniques. Okay. Thank you for your patient listening. Any questions you do contact me. Stay blessed. Thank you.